right? We need to consider it as a failure product. So these are some of the overview. What are the topics I am going to discuss about you? And so you know that there is our ASU and H drug where it's having a sources from the plants, drugs, minerals, and animals. And as per the products available in the market, either classical formulae or patent or proprietary medicines. And there is a safety study and efficacy importance as mentioned in Sachruta as well as the Sharakasham with both documents were already mentioned in the answer time. There is an importance of the safety and efficacy studies. And why it is important efficacy, because the patient acceptance and the ultimately the quality control role is, if it is a producing a efficacious product, then only quality control also will be valid. Otherwise it's not going to be a, efficacious. But most of the times we will not look about the efficacy point of view. Even if you are going to the field, many drug inspectors are there. If you are going to the field where you can't able, you are not focusing on the efficacy point of view. And as per the our objective of the PCIMH, if you are looking, there is a 4 5 objectives are mainly. So the publications and revision of the ASU and formulary, and one more, there is a major aspect is there is a drug research. So the drug research of ASU and products, and there is a also frequent publications. How there is a pharmacopoeia publications is are available, and similar manner where there is a other scientific studies. This drug research perspective, it is involving in all other organization also. Comes under the where the work come under the Ministry of Oil, example research councils and its periphery things. But also PCMH also having same kind of the role. And if you are coming to, there is a life cycle of the any medicine. In allopathic case is entirely different. There is a synthesized active pharmaceutical ingredient, and like that way. So if you are coming to the in case of the herbal, there is a one is there is the production of the raw material. Mainly there is a plant sources, then processing of the raw material, manufacturing, then you shifting to the manufacturing unit, then storage and transportation. Then ultimately, once if it is a manufacturing, it is happen place, and storage if it is taken place in the shelf life where it is a playing a role. And where ultimately next to the stage is where the customer. So the benchmark, what are the benchmark it is present in that is raw material production where there is a good agriculture practice is existing to control the quality aspect. And processing of the raw material where there is a field collection process also, it's where playing an important role. And if you are coming to, there is a benchmark of for the manufacturing medicines where there is the pharmacopoeia is the playing a role, and the GMP. These chapters, everything you have already seen. And apart from that, if you're coming to the storage and thing, ultimately there is a self life is playing a role. And next, the final stage, if it is coming, there is a public as well as the customer satisfaction point of view, safety, efficacy, as well as the adverse drug reaction and other product related issue, including there is a package, leakage of the package, and any other suppose where there is many other time you may be noticed in the field, the ingredients, the name it will be there, ingredients will be different. And where analysis time, even you may be seen that there is a chemistry section, they may be detected there is a diclofenac is present. So all these are where it is coming to the area of the last one, public and customer satisfaction or patients acceptance. So to play this, where there is a pharmacovigilance system, ADR aspect, and other product related issues where in pharmacovigilance you can able to report it. And if you are coming to in case of quality control aspects, PCIMH is playing a role. And what are the organization is mainly playing a role in this is the NMPP QCI is Quality Control Quality Council of India, and NMPP is playing a role in case of the production of the raw material and processing of the raw material. Manufacturing medicines, where if you are coming, there is a Drug and Cosmetic Act. Most of the things, if the issues is comes, it will be referred by the drug policy section of the DPS, our Ministry of Ayush, will refer to that of the Pharmacopoeia Committee and Scientific Advisory Board of the PC. So main role where we are playing in the manufacturing of the medicines also, you may be here that even the self-life stability study, everything it will be all things vetted and committee passed, then only it will be in the gazette notification, it will be present in that. So the public, all these areas were well taken care. If you are coming to the public and customer satisfaction, safety and efficacy not only meaning of only the mercury level, or in case of the heavy metals level analysis or aflatoxin analysis, that is a one of the contamination point of view. And if the contamination, if it is a more, where the safety issues will arise. Apart from that, if the specific drug related intake, after the intake, what are the safety issues are, that is a much important. Although the quality control is played in the major role in this area, but after the intake of the drug, to what kind of the safety aspect is things that is that will vary from person to person, because genomic will definitely playing a role, and there is the conditions of the shelf life of the product also it's playing a role. So the safety, real safety aspect means where that is the safety aspects will be the playing a major role. Then efficacy, if the product is whatever product you are giving, if your product is not a efficacious, then no need of the making a quality control aspects. That is totally it's a waste of the things. So if you are coming to the quality control prospect, there are many things where you may be seen that you have discussed many of the aspects. Now we are coming to the biological point of view, toxicity and efficacy point of view. I am going to focus. And in regulatory framework, where the pharmacology is playing a role in the quality control aspects, or in case of the standardization point of view, where it is playing a major role. You already last 3 days you have seen there is a various section of the Drug and Cosmetic Act as well as the rule. 3A about the AAC and 3H about the patent and proprietary medicines. And if you are coming to the chapter 4A, where chapter 4A it is mainly in case of the provisions related to the AAC. You all well aware of the things. And main thing is there is a schedule. One authority books is playing a major role in uh, the content of the pharmacopoeia, because whatever the formulation or it is available in the authority books, we need to bring into the pharmacopoeia. And this authority formulations, the books, whatever the formulation it is existing, that formulations if they were using in the market, if for that standard only we are pharmacopoeia will be published. So that is very important, the first schedule. And in case of the second schedule 4A for provides the quality control of the homeopathic test. Apart from that, schedule E1 also it's a playing a role in the drug licensing point of view, because schedule E1 it is mainly dealing with that of the poisonous substance like a cannabis and such a kind of the even that also it is used in the certain kind of the formulation. So the licensing point of view and safety aspect we need to taken care of that also. And if you are coming to the major aspect of the things, what is the chapter 3A is saying, already you have 3 days you came across everything. So keep in the underlined point, it is a manufactured exclusively in the accordance with the formula described in the authority books of ASU. Then that is in the first schedule. The next one is the patent and proprietary. And what are the other kind of the things it will be mentioned, that parental route of administration, it is not acceptable in the things. And other kind of the things where if you are coming to, there is a positive health promoters, there is a poshak balya, so unavu porukal in case of the siddha. So all these things where it is already it is mentioned in the first schedule. And if you are coming to another one is where is the cosmetic alagu sadhan, Hassan. So all these things will be also in the first schedule. As such, if you are giving the formulations, there is no need of the any the easily you will get the license. The new one aspect point of view, if you are coming to, there is the medicinal plant extracts. It may be a dry or wet extract also. Mainly there is the aqueous and hydroalcoholic we will use, because most of the time in case if you are going for the preparation of the formula. And in our traditional system, mostly there is a aqueous and fermentation process that is taken place. That time where there is a algal protection will be there. So hydroalcoholic algal, if you are going, there is a no need of certain kind of that studies. There is a exceptions will be present in that. And these are the 158B is a rule. It's playing a major role in case of the licensing point of view. So these already available in the CCRS guideline and everything. And I'm coming to directly to that of the research perspective. The same thing also I have reproduced here. Research perspective, what are the things it will be present. So if you are going for the licensing point of view, the classical ASU drugs defined under the 3A Drug and Cosmetic Act. These are the things. If you are giving a first one, if you are giving there is a text, ingredients are as per the text, indications are as per the text, and the clinical non-clinical and clinical data is it is not required. And if you are coming to in case of where there is the another one, if you are coming to that of any changes in the dosage form, suppose if you are in the churan form, suppose if you are going form or capsule form, no need of the any kind of the studies, but the ingredient should be as per the text. And the preparation of the things also, the formulation also as per the text. And if you are coming to where there is a requirement of the studies is arrived, if you are bringing the formula as such in the things, but if you are proposing to the new indication, for example in corona time we have the many of the things are in case of the chikungunya fever time where we have the different thing. And if you are coming to that cut of where the formulation will be useful for the new perspective. So that time where there is a proof of the clinical, mainly there is the non-clinical and clinical data. It is required. Actually, it is a both. The sixth column also it is the clinical. Sixth column is there is also the clinical perspective, but I have where it is a non-clinical mention. You can omit that. Then patent and proprietary medicine. Next one. Everything it is the same. Where if you are bringing with that of the alcoholic extract, aqueous extract point of view, where that is also patent propriety. But the formula, if it is as such, production it is as such, there is no need of the studies. There is no need of the data it is required in that. But if you are going to, other than the aqueous extract, if you are going to make in the patent and propriety, where there is a non-clinical and clinical data, safety data it is required. So safety data, if it is required, it will come single dose to oral dose, single dose toxicity to repeated dose toxicity, and everything it will be applicable. Similarly, if you are coming to that of the, in case of the depends upon the preparation, if it is a oral route, you have to make the thing. And in case if you are coming to that of there is a topical route, where you have to apply the topical toxicity test also. And similarly, in pattern and property, suppose if you are going to make any kind of the E1 point of view, there is a schedule E1 ingredients if you are adding the cannabinoids, all these things, if you are adding in that, so there is a toxicity and clinical efficacy thing is required. So if so coming to the nurture, where if you are coming to where, what are the things that is required in this point of view, in our India, as per the Drug and Cosmetic Act in our country, the herbal manufacturers following ASG pharmacopoeia after the quality control and the ASG medicines, the safety and efficacy studies are not required, because if you are, they were following as such a classical formula, because it is a well established, we are using from the ancient time itself. And where if you are coming to that of we have exposed the PV program, pharmacovigilance program, and we need to more effectively we have to look that. So if you are looking, there is a lot of safety issues and product related issues, because in the PV program, not only there is only ADR, you have to report all the things. There is a package point of view, ingredient, if anything is mismatching, or if efficacy is the non-efficacy of product is very important. You have to uh, you have to tell to the retailer, as well as in case of the patients, or if the physicians also need to be educated about the non-efficacious. There are many non-efficacious means non-effective formulations. Nowhere it is mentioned in case of the in our ASU and drug. That is also, if we are bringing that, then only we can able to check it whether the problem is with the quality control, or really where is whether the ingredients, same ingredients, we can go to the backlog work, whether the ingredients are same as per the classical literature, they may be given in the container, and whether the ingredients it is present in the same, and how we can able to, whether quality control aspect, any kind of the defect is there, we can able to manufacture, look at that manufacturing issues also. But if you are going to the international aspect, pick safety and efficacy studies are, it's a very important point of view. We need to bring our product to the international market. And but international market also, there is a PV program, it is already existing. Now if you are coming to the pharmacology, where the pharmacology is linking, although quality control if you are saying, and where if you are coming to the pharmacology is linking, and what is the importance of the thing. So role of pharmacological studies, and what kind of outcome it is going to provide. We have in allopathic drugs all the aspect we know. So similarly, we want to establish our the drug system more acceptable by the patient, not only in domestic as well as the in case of the international aspects, if you want to make it the things, and ultimately which will make there is a more industry growth, and where the economic review of the economic level, where it will get increase in our country. And in such a case of the time, the pharmacological studies it is important, and we need to bring there is a certain kind of how the product is working. However, Ayurveda system, ancient system, already they written, they documented, and we need to go to the backward reverse pharmacology aspect. We need to go through it, and we need to make it that yes, our the formula which is prescribed in the traditional books, it's acting through these mechanism, and hence the these efficacious for our any kind of the disease condition. So scientific based approach we need. So we need to go with instead of from the in silico, in vitro, in vivo, where we need to go to the top reverse pharmacology aspect, we need to go through it. From clinic already it is existing, the formulations it is used, we need to bring to the top in silico, in vitro, then how it is clinically it is affecting, we need to make the correlation the aspect. So mainly in case of the pharmacology section, it is focusing on the two kind of the aspect. One is the reverse pharmacology aspect, another one is there is the system pharmacology aspect. I will explain you later slide where the system pharmacology point of view. So reverse pharmacology approach, as you know everything, the central thing is traditional herbal medicine in orange color, where you can able to look that central one, traditional herbal medicine. There are different way we can able to move. And anyway, if you are bringing into that kind of the what we are normally doing, if you are looking that there is uh, there is a left side, the quality control and standardization we are doing well. And but reverse pharmacology work, now there are many places we have started. So experimental documentation, exploratory study, laboratory research, then again we need to confirm it. Suppose in case of any other there is a full core medicine used. There are many medicines that they were using that. So if you're coming to that of where there is the full core use medicines, where we need to make it as a standardization point of view as well as the clinical effect point of view. And what is a new drug? There is a possibility. Suppose if you are going and taking a traditional medicine, we need to focus. There are now there is a reposting of the molecule also. It is available. The best molecule we can take, whether we can able to other disease, whether we can able to use the same traditional medicine if it is they were using in other diseases, we can extract bioassay guided fractionation. We can able to make a different extract. We can able to analyze and which extract it is that the molecule isolation. Then we can able to bring into the case of the new drug. So that also will make in the things. I will tell you how you can able to connect this with that of the quality control aspect. That is the final thing. We will come to that where the pharmacology role also playing in the quality control aspect. Another one is there is a reverse pharmacology. Another one is there is a system pharmacology analysis. Once the molecule if it is enters into that of the our system, how we know that in case of there is a allopathic drugs where it is acting, what are the what are the biological pathway it is going acting, and how it is producing effect, whether it is suppressing some kind of the processing inside of the things, and how it is reduced. For example, in case of the pain, COX-2 inhibitor, celecoxib, broficoxib, where we are using, or ibuprofen we are using. And how this ibuprofen acting, we know that it's a COX-2 inhibitor, and hence where it is reducing interleukin and anti-inflammatory thing. So similar manner here also we need to focus in the system pharmacology aspect. There are system pharmacology aspect in the traditional medicine of Chinese where the very commonly they used, and where this is one example I am showing here. And similarly now we are also trying to we are also working to make our evidence for the thing. So if you are coming to the traditional medicines of the Chinese medicines, the example where they taken the molecules, they did the in silico, then there is a molecular pathway in vivo, and followed by where there is a how it is acting in the things they made. So there are some of the examples where I have mentioned. So we will come to a similar manner. We have already worked in our lab, dry lab. That also I will show you. So first, what they were doing now, they were going to take the any of the database. They worked with the database, but what we will do, we will take with a particular plant which are mentioned in the authoritative books. Example, if any of the uh, classical formula is there, 5 plant is there, out of 5 either one plant or the 5 plant phytoconstants we will analyze in a manner, and we will analyze, we narrow down it, and we will go to the in silico study, in vitro, and in case of the in vivo study. So what they did, they analyzed the compounds, q enriching herbs where they analyze the active compounds, and they target analysis, network pharmacology. There also it's a software oriented work. We need to identify the new network, or where the acting target, or in case of the already existing one. So network pharmacology work we can do that. And how there is a, we know that there is a traditional medicine, the one classical formula it is used for the many diseases, correct, to treat in the treatment of many diseases where they were prescribing. And how it is working, and what are the different kind of the interactions it is taken place, and what are the different kind of the target. If you are looking at literature of the any of the medicinal books, Indian medicinal books, they mention the same xy drugs, where it is mentioned for the diabetes also, epilepsy also, you can able to notice. Even it can be used for other inflammations and the hepatoprotective activity and all the things. How it is interacting with things, we need to go and study the aspect. So these are the active constituents where they identified, and they made there is a what are the target genes there it is there. Similarly, another one is there is a blood tonifying agent, and they proved finally how they effect is producing. So similar manner we need to document for our traditional system also. And apart from that, what we are going to do after the documenting, how we can link to the of them in future, if the day by day where the growth is important and revolution is important. So we are going to nowadays, now it is not possible. In future, if you are making some kind of the database, and whether we can able to bring the quality control system much better, and where we can able to relate that quality control system of the phytoconstituents with that of the efficacy point of view, correlation point of view. For example, if you are looking in case of the paracetamol, one small example, paracetamol. If the paracetamol, if it is reducing a fever, so if you are going for the assay, yes, 95 to 100, there is a range, 110% where the paracetamol assay, where it is there, pure drug it is there. Because of that, suppose if it is a 65 percentage of the assay it is coming, the drug is will not be efficacious, will not produce. Similar manner here, but only thing is we need to concentrate here on the multiple compounds, phytocompounds compared to that of the single compound. So that's a major challenge it is existing in that. Okay. Now quality control aspect, how these evidence based pharmacology studies are going to be used. And as per the WHO quality control in 2017, they published 55th, first there is a technical series. The quality control drugs, including herbal drugs, based on availability of the marker and reference substance. You may be heard about the botanical reference substance and all these things, phytochemistry reference substance. To identify the active marker or reference substance for the herbal drugs, it is mandatory to isolate the molecular with biological activity. So if you are coming to the marker compound, how also the marker compound, biomarker compound also, it should be easy to that of the analyst also. The analyst should not find any difficulty to quantify the things or to identify the identification. They should not find any analytical point of view. They should not find any trouble. So these are the main thing is there is a marker compound should be identity, specificity, and selective using a specified analytical techniques, and traceable quantity should be available, easily detectable, and easily obtainable and stable. Such a kind of the compound we have to do. So the WHO has proposed there is a three kind of the thumb rule. One is there is a for the quality and safety efficacy aspect in case of the herbal drugs for the quality control aspect. Rule number one is if constants noun therapeutic activity or activities have been identified, that should be the first compound should be you have to use for the marker actual point of view. If it is not available, next one, suppose if the one is not possible, then at least constants which are recognized to pharmacological activity, either preclinical studies or any other kind of the studies where if you recognized, that should be used as a marker compound. But third rule, if everything both it is not possible, then we have to use some kind of the chemical marker, analytical marker. But most of the time we are very few plants we are using we are applying a rule number one and rule number two. If you are using in case of the curcuma longa, curcumin, mostly we will consider that as a common. In case of Withania somnifera, withanol, where we can able to consider the thing. But the many kind of the things where we cannot able to correlate with it, correlate with the kind of the things. So mostly we are using the chemical kind of the marker. So there is a pharmacological activity will give clue and identify the particular marker which is responsible for the activity. So we can able to at least we can able to bring that thing. So as per the rule number two, we need to identify the marker compound and recognize the biological activity. So pharmacological active markers can be used in future. There is the herbal control quality aspect. So outcome, what it will be the evidence-based studies, reverse pharmacology or system pharmacology will help to identify the molecular mechanisms, how it is acting, and pharmacological active compound will be the therapeutic in future. If you are bringing any kind of the pharmacological active preclinical study, if you are finding some of the chemical molecules, phytoconstants which are active, we can bring that develop into the clinical marker. And fourth one, where there is a strengthening of the quality control aspects we can able to do with the thing, and strengthening of the scientific evidence on the molecular mechanism and safety and efficacy also, where we can able to establish the thing, and positioning of the herbal medicines, ASU medicines to the top in industry, in the international level also, as well as the domestic level also, where there is a more acceptance of the patients, as well as the product confidence also we can make, and which will also give to the market share also the economic growth also will be there. So what kind of the activity pharmacology section is doing here at present? In silico pharmacological studies, molecular docking and mechanic simulation studies we are doing, and network pharmacology also there is a part of the things we are doing. And in vitro pharmacological studies, especially we are not doing especially we are doing only there is the enzymatic inhibition and kinase study. The lab is it's under development. I think next another 6 months it will be get ready. And in case of the cell line study, we will make into the top of outside sources we will make. And next one is where there is the in vivo final. There is the in vivo pharmacological studies with the molecular pathways, with using there is a modern advanced biological molecular biology studies using the Western blot, PCR, and what kind of the things it is happening in the genetic level, gene level, whether if anything it is happening. So for that in vitro, in vivo studies for the zebrafish lab also it is in the establishment, and hope another next financial year it will be get in the way. So these are the some of the references where which I gathered. And now, although I said that what kind of work in silico work be done to prove our system, our medicinal plants, some of the medicinal plants for the efficacy, pharmacologically it is active. And these are the Punica granatum, it's the pomegranate most commonly we will use in our day-to-day life. And where this paper has published presented in the oral presentation recently last month in the Jamia Hamdad International Conference in Traditional Medicine and System. There is a, in case of the cardiac system, there are many mechanisms. Is there beta 1 receptor, where there are many drugs it is acting in that similar manner, where sodium potassium ATPase is a it's like a pump. It is a very important one, where which inhibition will makes the certain kind of the calcium ions, mainly calcium ion, will be in increase, so our heart will be function more. So punica granatum, where it is used traditionally, we have noticed that punica granatum, where it is traditionally mentioned in the Ayurvedic books, the seeds of the punica granatum, it is having a cardiotonic activity. So we have done the thorough literature search. Till date there is no data is available in this aspect. So similarly, what I have explained, how the traditional system of the Chinese, they did a similar manner. Now we are also there is a reverse pharmacology aspect. Now we are going system pharmacology later we will do within, once the lab it is established, then we will go to the system pharmacology. So there is no proof for the thing. So what we did, these are the things we done the literature search of the punica granatum, and whether what are the phytoconstituents are present in that any cardiotonic activity is reported, and whether in silico activity is reported, and in silico activity is reported, whether uh, what is the ideal target for that thing. So molecular docking we want to do, what is the ideal target for the things, which kind of the protein, whether the protein is already isolated, or all these things where we have identified, and molecular docking we have done. And molecular, once if the molecular docking is done, molecular docking is a very short term, how the molecule is interacting with the enzyme. We may be studied in our biochemistry or plus one plus two level, where there is a lock and key mechanism in the enzyme, and how the molecule lock and key mechanism it is happening enzyme, how it will be translate into the kind of. So similar thing in the molecular docking, where the will bind but in a very certain time. But if the binding in a very slow time with or reduced some short time, will not make many changes in the thing. So how there is a binding ability is there inside of the receptor or enzyme, whether really it is the binding it is taken place, and how there is the binding if it is taken place, what are the all we know that all the proteins are it is made up of the amino acids. So what are the amino acids are interacting with that. So and how finally we are coming to the conclusion, whether that traditionally it is mentioned, whether it is proved in that tongue. As per our literature, if you are looking, there is a punic acid is a major pomegranate seed oil. It is were isolated, and it is reported for the anti-inflammatory and antioxidant, anti-diabetic, and cardiovascular benefits. Also, it was mentioned in there is evidence studies already reported, or these are the studies. And oleic acid, linoleic acid, all these things it is present. Quercetin, camphorol, luteolin. But if you are, we have shorted out, there is a number of compounds, and all the compounds where we have done the docking study. So our aim is mainly to evaluate the sodium potassium inhibitory potential of the phytoconstituent of the seed of the phytoconstituents. Mainly in case of we have chosen only 4. I will show you all the docking results to climb to prove that yes, traditionally what it is mentioned it is correct. So if you are coming to that kind of the materials, there is a this is the how we are doing with the in the software. So first we need to predict the target. The protein need to be identified. Protein ID 3A 3BY. I will show you the remaining things. So you will come to know the result. I will directly will come. Already I have explained the docking result. So if you are coming to the okay, sorry, I have not saved the other things. We have done almost 20 compounds. We have docked out of 20 compounds, where we have chosen only these compounds for we have done also molecular simulation study, but the presentation point of view we have chosen only these 5 compounds, and how it is making. So if you're coming to the docking score point of view, camphorol is showing a much higher docking score. Always the values will be the minus. It is a higher docking score it is showing. Where if you are coming to in case of the Obain, there is a reference standard drug. The Obain is the allopathic drugs, where it is acting directly on the sodium potassium ATPase, and where it is increasing inside of the calcium ion level inside of the heart, sacroplasmic reticulum, and where there's the heart contractility is increasing, so it is producing a more cardiotonic activity. So if you are coming to the point of view of in case of Obain, it is showing a docking score of -6. So it is a lower value. Other phytoconstituent, it is showing a higher docking score. Whether this docking score, again, we need to molecular mechanics, MMGBSA. Molecular mechanics is generalized bond surface area. How in the protein surface area, how the binding it is taken place, we have analyzed the MMGBSA. So where we come to know that although camphorol, it is showing a higher docking score, but if you are coming to in case of there is a binding ability point of view, where it is showing a lesser. And quercetin, it is showing a catechin, where it is showing a lesser binding docking score, but where it is showing into the mmgbsa, it is a higher. So which means that the mmgbsa, it is ensuring that the docking score, it's not a correct identification. You have to take that how there is a molecular inside molecules inside how it is behaving. So based on this, where we have chosen all the docking things we have made. And always we know that once if the molecule goes, we maybe studied that there is a different kind of the rule. It's a drug like substance or not, all the molecules will not produce effect. There is a certain kind of the rules is present. For example, molecular weight could be less than 500 or 400 molecular weight. Mostly that kind of the chemical molecules will be easily absorbed in our body. Similarly, how much hydrogen bond is there, hydrogen acceptor bond is there, donor is there, and log p, whether the molecule is easily lipophilic in nature, whether easily, because our body is made up of the more of the lipids and things, so whether easily absorption will be taken place. All these things will be we have studied through the software. And Chemforol, it is showing a molecular weight of 448, so which is it is as per the Lipinski rule, it is fails. But some of the place where it's showing activity also. Now if you are coming to, there is the structure which is showing in the folded manner. This is a 3A3YB, that is a human protein sodium potassium ATPase, isolated from our body, crystallized in the human source without any mutation, with the binding of ovine. There is a standard reference drug with that. So now if you are looking in this, there is the ovine is binded. Ovine, what are the amino acids it's binding, where we have circulated in the thing? There is a glutamine 786 where we have made the circulation thing. And if you are coming to the other, this is a phytoconstituent. There is a synergistol where how it is binding. And if you are coming to Chemforol, how it is binds, and catechin, how it's binds. So it's a simple interaction point of view. Now we need to confirm it. Whether the binding pocket, this is the where the surface view of the protein, ovine docked pocket, where it is the ovine is docked. In the protein, if you are looking, there is the many pockets will be there. Whether the same pockets, whether drug is binding the standard drug, that is the one of the conclusion, where if you are looking that the ovine will be the same pocket, then remaining all the protein, all the molecule also we are talking in the same place. So which we are confirming that already established drug in the market, which is doing a well in our human, where it is showing the efficacy is, whether the same binding pocket, our phytoconstituents also, whether it binding, where it can able to produce a activity. So if you are coming to the things, these are the amino acids where the interaction taken place. Ovine reference drug, where if you are looking most of the things, there is a glutamic acid where there is a playing a major role, and catechin cousin also where it is acting in the same place, but synergisol it is not showing. So which seems that if you are coming to in case of the docking score, if you are looking the synergisol, it is showing a -4 value, and if you are coming to mmgbsa also, it is showing a lesser value, 27 -27, which confirmed by docking studies as well as mmgbsa, the result has confirmed. Yes, because of that reason, where there is a common ovine, where it is binding, it is the glutamic acid. Where if you are looking at catechin and things, it's binding in the same interaction, it is taken place in the pocket. But the synergisol, if you are looking, it is not having any interaction with the glutamine 786. So which means that it is not having any in the pocket. It is simply it is sitting, and it is not having any interaction. Whenever there is a hydrogen bond forms between the molecule, you know there is a chemical structure where there is a OH group, NH2 group, all these things will be present, where it is mainly playing a interaction point of view the protein. So to confirm that how this binding ability, we have run the molecular simulation study. Ovine, how it is binding. The first one, if you are looking that the blue color is the protein and red color is there is a ligand. So if you are coming to in case of the similar the binding ability, where will be how it's a binding ability will be analyzed, how there is a stability is present in the left one, whether the if zigzag trajectory, more it is a trajectory, since there is a, here I have stretched one the figure, you cannot able to see the straight line. If you are viewing in the full thing, single point, single the graph itself, where you can able to notice there is a where there is a straight line of the trajectory, you can able to notice. So now if you are coming to that, what are the interactions it will be present, where you can able to notice that there is the green color is the hydrogen bond interaction, the hydrophobic ionic water. So these are the amino acids where they interacting with the things. So similar kind of the amino acid interaction is there also. We are going to look in that in the quercetin. Quercetin, if you are looking at where you can able to see certain kind of the straight line between the 40 and 60, almost 40k, thoda other there is a 50 to almost 65, where you can able to look the straight line, and further there is the 65 to where 80, where slightly you can able to. So which means that these area where there is a binding ability is more. Similarly, if you are coming to in case of there is a catechin, you can very clearly you can look at the initial interaction very straight from around 10 nanoseconds to around 40, where it's a straight line. If the trajectory is similar, there is the major things, which means that where there is the binding of the molecule with that of the protein, it is a very good binding ability. And slight trajectory has increased again, where if you are coming to almost 50 to almost there is a 80, where the straight, so which means that binding ability is present in the more. Similarly, if you are coming to the camphorol, but now if you are looking the molecule where there is a, which are the amino acids, how much percentage of the amino acids were interacting, isoleucine 392 or 97 percentage. So which means that docking score, where you will simply you will get a amino acid, but in case of the simulation study, you will get how much percentage of the each amino acids interacting with that of the molecule. So these are the percentage of point of view where I have mentioned, you can able to notice. And finally, what is our conclusion we are making? Punica granatum, although, okay, I forgot to actually, if you are coming to the thing, I will show you the only thing. So if you are coming to the of this docking score, if you are looking as per the docking score, Quercet camphorol is the highest value. But as per the MMGPSA, where there is a camphorol is the lowest one compared to the. So we can now we are going to make with the only of the MMGPSA result. We are going to compare. But quercetin and catechin, if you are looking, although there is the lowest docking score, where MMGPSA binding, it's having a highest thing. So which means that the binding ability with the inside of the molecule, it is a much more. That is again confirmed. If you are coming to in case of the synergisol, although there is MMGPSA, it is a value is 32 only. So that was again confirmed here. If you are looking camphorol, it is having a only one interactions, where if you are looking catechin and things, the percentage of point of view, if you are coming, the interactions will be confirmed. So which means that based on these conclusion, MMGBSA and molecular things, we are coming to the conclusion that Punica granatum, quercetin, catechin, it is a polyphenolic compound, it is present, where in silico study, where we found that it is having a stable binding with that of the sodium potassium ATPase, where already there is a allopathic medicine reference standard which is used, already proved in our system in the same binding place where it is binding, and hence where it is having a possible mechanism to produce a cardiotonic activity through in silico study. We are confirming that sodium potassium ATPase inhibition. So we want to in silico weighted, whether we have gone through the literature, whether already it is the punica granatum is or quercetin. Now we are confirming that quercetin, catechin, whether these has reported any kind of the cardiotonic activity. So in 2015, where we found that there is a quercetin has reported to increase the cardiotonic activity in the isolated cardiac myocyte. This is a in vitro study, isolated cardiomyocyte from the mouse heart. They found that it is increasing a calcium level inside of the heart by it's showing a cardiotonic activity. So which mechanism possibly we are confirming that? So they also our study, what it's confirmed that it could be there is an inhibition of sodium potassium ATPase. Further, how we can able to make evidence of the thing? Now we need to go to the in vivo study or in vitro, any kind of the cell line study or in vivo study, and we have to isolate the heart tissue or something. And sorry, heart tissue, we have to make the things, and we need to the gene, which are how there is the expression is there, where with the Western blot molecular techniques we have to analyze and different groups clear. Now we need to explore, and we can able to come to the conclusion study. But at present study, there is a in vitro in silico study. We are confirming the in vitro result. In vitro already 2015, where quercetin has reported for the cardiomyocytes increasing activity. So we are proving that this in future. Suppose if you want to make, although quercetin, catechin, in punica granatum, what are the marker compound we are using in case of the quality control aspect? So whether we can able to bring this compound as the marker compound, so that will be debate, and it's keep on. It will be rolled with the committee and all the things in future if you are coming to the things. But most of the things what they were saying, even one question also they raised by the our in the presentation time, quercetin and catechin is a very common compound, and how you can able to choose that. But actually we done for the 15 compound, we have taken for the presentation point of view only two. We have already run the many other compounds. We are going to do the publication. So at the time of publications, apart from the quercetin and catechin, and we have other compounds also which are specific to that of the punica granatum. So punic acid and such a kind of the where we have the compound. So that compound we can able to use and more. But apart from that, what? Suppose 15 compounds we have docked. We got the 8 compound which is having a good docking score and mmgv, and 8 compound we are running, and multiple compounds it is acting in the things and different mechanism we are going to study. So this is a one mechanism. Apart from that, any other mechanism, whether some of the compounds also will act on the beta 1 receptor, we don't know. So in cardiac, in heart, there is a lot of targets are present, receptor present. So we are having things that the multiple compounds will act in the multiple mechanism point of view. So that's the thing. Our traditional system of medicine may have whether synergistic activity is there or not. So that also we are going to confirm by the pharmacological studies. So which ultimately gives that proof to that of our traditional system, and very confidently to use the things and confidently to speak to them. Yes, this is a mechanism. This is the way where it is acting. These are the constant it is present. Only thing is in quality control aspects, we want to measure how much percentage of the compound or quantification of the compounds we need to do the thing, and we need to correlate. So that work it will be go to the chemistry point of view, but we will conclude that these are the phytoconstants it is making it there. So we are pharmacology section is a playing a role mainly to make evidence. We are going to create a scientific evidence to our system and phytoconstituents level and molecular level and future our system medicines we are willing to bring to that of the international market also, and in quality control aspect also, like a WHO guideline we are going to link that, and it will be a more robust manner kind of the quality control in future we may be expected. Thank you. So if any questions you can.
Sir, normally which software you're using here for docking?
Soringer.
Student.
We are using because we have seen there is a lot of variability between the auto dock and things, and even 2012 onwards I am working in that. I know that there is the same thing, the compound or protein if you are giving to one person, another person if it is working with the free software, available software, the protein preparation is ultimately there is a defect, the result will be vary. So that's the reason we are using only Soringer software.
So chemiferol and this quercetin is already available in this library, hopefully.
Huh?
Because I'm also working on quercetin
uh,
for some other activity. Uh, but I think that the docking data and everything is available on that software.
which software?
Stranger.
So
Nee. Okay, sir.
we,
Maybe.
we have searched the literature, we done the thorough literature search, and we have means it may be available for other target,
Maybe. A
it
lot
may
of
be
options.
available for the other disease target. For example, if you are going for anti-diabetic activity, it may be available.
Maybe, sir. It's
In sodium potassium ATPase specifically, whether is it available means that probably may not.
okay,
As per
sir.
our literature search, up to last month it was not there, because literature search will vary with after next day if you are doing with the thing. Suppose if any the data if it is
Sir.
uploaded, it will vary.
It's a word of science. Thank you,
Excuse me,
sir.
sir. Sir, is there institutions in the South India they're making the molecular docking and all?
So maybe there are many institutes they were working, sir,
Yes,
but
sir.
it's depends upon the who is working.
Yes, sir.
How they were projecting, how they were implementing, and how they were selecting the things, and that's entirely it's a different aspect. The view of the scientist will vary from one person to one thing. The data also, the result also will get vary.
Sir, one doubt, sir.
Yes.
Sir, whatever we will involve bioinformatics tools. Uh, Finally, we will match our in vitro result and
Yes.
in silico means sometimes it is not matching now, sir. In vitro, sorry, in silico it is prediction now, sir.
So that's why we need to use in silico as a prediction. But this software which I am using, it's an industry using software. All the pharmaceutical, most of the throughout India, it's not only the globe, most of the things they were using the software.
Yes, sir.
So they already 70%, 80% results are matching.
Yes, sir.
Okay, so that's the reason I will no, I need only this software, although it's a cost,
Yes,
very
sir.
very costly. 1 year subscription it is coming 8 lakh with the molecular dynamic things, 8 lakh with only 1 year subscription. That is also only number of token is very less,
Okay, sir.
only 10 token, 12, 15 token. Below that, they will not give.
Yes.
Below that, if you are taking also, it is a waste. 15
Yes.
token ka niche, if you are looking, the software will not work in a proper way.
Yes.
Okay, so the very few, the cost is more. So precision also is there.
Yes.
That's the reason we are taking.
Yes.
That's the reason I am not also using the other free software.
Yes.
So the data almost mostly it will come if you are choosing a right target and rightly if you are making a thing, and it will come. So you have seen, I did with that. We did with the in silico study.
Yes, sir.
And 2015 already where isolated cardiac myocytes, it's a reported cardiotonic activity.
Yes.
They mentioned that increasing a calcium ion, and how the calcium ion will get increased. So that's where the sodium potassium, the pump where it will become in that mechanism point of view. But we need to also ensure with the other things. Any other mechanisms, whether it is playing a role, also we need to ensure it.
Yes.
And we have other target also we are working, but it's a time consuming process, because
Okay.
if the MD, if it is molecular dynamic, if you are running, actually we need to, nowadays the journal paper are accepting minimum of 200 nanoseconds to 300 nanoseconds, but we
Yes.
are run to only 100 nanoseconds. But 100 nanoseconds it will take 2 days. Continuous power supply is required.
Yes.
And many times we face the problem that if you are after running a thing, so one day and the next day if you are looking, it will get, is the power failure is there, our system automatically goes, then again, so that there is a lot of things is also is there in
Okay.
that. So, but mostly it will come if you are using this software.
Yes.
We can able to get almost kind of
Okay.
the results. If it
Yes.
is not coming, then we need to relook that, apart from this mechanism,
Yes, sir.
any other mechanism, whether it is acting or not.
Yes.
So target we need to change, because in our literature they mentioned our
Yes.
our aim is to make a evidence. So we are focusing now only sodium potassium ATPase. If the sodium potassium ATPase target, if it is not working, any other target, whether whether it may be
Might be
working.
work.
And suppose quercetin is not working, after that there is another 8 compounds is there. Actually the data I prepared, but sorry to I
Yes.
didn't
Yes.
copy
No.
the thing.
No problem.
Actually last minute में I prepare कर दिया.
Okay.
Sorry,
Yes.
I have not included I have not included that. Actually deleted. I will see that. Any other question?
Sir, I would like to ask one. You have selected Punica granatum seed, which is a single plant material. If we have a multi-ingredient product, sir,
I am
yeah,
listening, please.
yeah. So if we have a multi-ingredient, as you
Yes.
have done in silico with, as you are telling, more than 8 compounds,
Uh,
in a multi-ingredient, how will you plan to do this type of in
that is a big
silico?
task. Next
Because
formulation we will take.
yeah, I need just idea or suggestion from your side. That's all.
See, we will work out that. I have planned that also. Main thing is now the software. I need to get it the things. So
No, no,
I
I
need to
just
go to what the process I did for one compound, one plant,
the polymorphism type it will be, I
one
think.
one 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 plant. What the literature says I did, I need to do with the five plant. Suppose if the five composition is present, five plant material is present in the thing. I need to do for the five, and I need to go through
Yeah,
out of the
yeah,
five. I need to narrow down.
that's okay, sir. But my point will be from the holistic approach that in a multi-ingredient, only few will be the more potent according to the formulation for a particular use. So if you take all the compounds, it is again waste. So few particular indication, few ingredients will be mainly responsible, and that composition again will differ how much quantity they are adding and how much we will get it in the in vivo study. So accordingly I need some suggestion from your
So
end.
that is the where the docking score is playing a role.
Okay.
Apart from docking score, where again if you are coming to reconfirm the docking, see docking score. What is the meaning? We are short outing the molecules. We are short outing the molecules. For example, now you have seen the sodium potassium ATPase. Sodium potassium ATPase. I have shown only for four compounds. Now you can see these are the sharing is possible. Huh. Actually, I prepared in PPT, but unable to bring that. Uh, Actually, you can look that there is a toxicfolane, cyanidine, all these compounds it is present. So based on the score, we will short out. First plan is based on the docking score, whether the docking score reconfirm करने के लिए where we need to do with the MMGBSA. MMGBSA, it is the score is different, then we will omit the docking score and we will choose that as per the MMGBSA. So because the binding ability is important, so target to target, as you asked. Suppose I have, for example, beta 1 receptor, I have sodium potassium ATPase, and other kind of the receptor is there. So each one, the binding ability will vary, that we need to assess. Then we need to pool everything, we need to sit and analyze our where wisdom things, we need to make the things and bring to the conclusion. Multi mechanism. Huge time, and there is a infrastructure also it is required. We are working on that. That we will see in future. We will bring that. We have all the things we kept for the things for the publication point of view. We kept because all the data we cannot able to make.
Sir, excuse me, sir.
Yes, sir.
Coming to the analogs,
Yes, sir.
both actions are same. Suppose curcumin prepared from organic material and synthetic curcumin is analog of that. Both are clinically same. Sir.
It will be,
Action.
if the structure is same, it will be the same compound, same structure. If they were making, and purity, they have to look that it will work in the same. There is no other option, because ultimately it's a molecule structure how is there, that is a playing a main role.
So there is no difference. There
Difference
may
will
not
not come, sir.
clinically.
Only thing is,
Clinically.
sir, in case of clinical difference, it depends upon the purity. Okay. Second thing is, if the chemical structure is same, the mechanism is example curcumin. Why they were going a synthetic mean? Mass production we cannot able to make from kind of the natural sources, and it's a time consuming process. And purity, to bring the compound in a more pure compound, 95 to 99 or 100, it will take some time. So instead of that, they were using the synthetic. That's all. But chemical structure, if it is the same, if it goes to biological system, it will, whatever effect it is producing, same effect only it will.
Okay.
But impurities are there, then it will be the issue. At the time of synthetic time, where the chemical synthesis they were doing, the impurities are comes, then definitely some kind of the adverse effects or toxic effects we can, it may come there.
Silico, how do you select a protein for a particular molecule docking?
Correct. So in silico study, see that purely depends upon the scientist. First thing, the background of the thing. Other thing, where you have to go to the normal step. Protein data bank, RCSB protein data bank is there. It's available free. You can able to search it. Whether net it will come. It will net will. It is there. So this is the thing. RCSB lead homepage. So you can go here, and there is all the protein they made the things. But you have, there is a certain criteria is there to select the thing. So based on that criteria, where you can able to select the receptor, many protein, all our body system in our receptor, everything if you are taking made up of amino acids. So that's we can generally we will call as a protein. So you can type any which kind of the things you want. This is name. So that will come. Then you have to sort out based on the certain criteria. You have to sort out the thing. You can download it, and you can able to make. Any other thing? That's all. Okay. Thank you. Hope I can able to next time if you are coming to, hope I can able to present with also in vitro in my study. So it will be more.
you need to go